So what I thought I would do is talk for about 20 minutes or so on telling you about Batten disease, telling you about the work that we've been doing as a consortium, sort of working together to try and understand and develop therapies, new therapies for Batten disease. So just some pictures to introduce you to the consortium. So first of all, we're an Horizon 2020 funded consortium. We consist of 14 partners from 7 countries, and you can see those coloured there in Europe. And that's it. So this is us at the beginning when some of us didn't know each other very well at all. A picture of us mid-meeting looking much happier and more relaxed, and then that was last September at the end of an international meeting, and we have our final meeting here in Barcelona. So as you can see, quite a number of us. So the whole concept of the project that we've called BatCure is to work along the pipeline of developing that's required to develop therapies. Now, we'll tell you a little bit more about Batten disease in a moment, but there are a number of different types, and we chose to focus on 3 types in particular that are caused by mutations, defects in membrane proteins, intracellular membrane proteins. So these are the most challenging types to develop therapy for within the BatCure field. So so the concept, as it were, was to spend quite a lot of our effort in terms of sort of the pre-discovery work that's required. So trying to understand more about the disease in animal models, in cells from patients, and also in very simple models that we developed. And also in patients themselves. And then our focus was to, we decided to focus on developing drug and gene therapies. So we already had some leads, etc. we needed to optimize and just further that whole process. So there's been a whole package of work around that. And then we also wanted to be aware of anything we could do in advance to prepare ourselves so that when we reach the stage of clinical trials, we could get to those as soon as possible. So that's what the last section was for. So, as I say, we've sort of the fairly standard pipeline in terms of translational research is listed here, some from our disease models through to identification of good markers to follow the disease and therefore to assess the efficacy of therapy. And then our lead development, optimization of current leads and drug and gene therapeutic strategies. So the different groups within the consortium already had some leads, so some small molecule leads and some background work on gene therapy. So whilst some of the aspect, we were starting here, other aspects of the biology sort of fed in partway through, and the thinking behind that was that we would make better progress building on things that we'd already gained quite a lot of knowledge about. Right, I'm not expecting you to be able to read this, but just simply to say that with a large group of people and having some quite complex deliverables and milestones required for the EU project, that we had to be reasonably ordered about it, very ordered about it, and mostly we've kept to the time. There have been some deliverables that have been delayed for technical reasons, etc. But mostly this was a real help to sort of keep the project moving forward. And of course we have an organisational structure to try and work out exactly where all, so that we can work along all the way all things fit in. So anyway, I shall move more onto the science now. So this particular consortium is actually one of a number that have occurred over the last 25 years across Europe. So there's a strong history of collaborating across Europe for Batten disease, and whatever happens with the UK and Brexit, hopefully that will continue. So this is quite an early one. So that's me at the beginning, looking much younger than I am now. That was NCL models, where we were developing some of the very first models. And then there was a DEMChild led by Angela Schultz here, which continued that work and had a real focus on setting up a disease registry for Batten disease. And then there's here's BatCure with a translational focus. And along the way, we've produced a number of books as well, which we class as reference books. So we've worked very hard to develop a good resource for anyone who comes into contact with Batten disease that needs something to dip in and out of. Such a rare disease. There's a lot of knowledge out there, but it's spread, and of course many people don't know too much about it. And by the end, or beyond the end of BatCure, we'll hopefully be producing the 3rd edition of this book as well. Okay, so back to the disease now. So Batten disease, its long name is the neuronal ceroid lipofuscinosis. It's a family of diseases, usually childhood onset. And the Batten comes from this gentleman here, Frederick Batten, who was a physician in London around the turn of the last century. So it's one of the most frequent neurodegenerative disorders of children, and in the UK, around 10 new cases a year. And it's difficult to know across Europe, but I've calculated that would make about 70 new cases across Europe each year. So these diseases have a characteristic ultrastructure of the storage material that builds up in the lysosome, which is also autofluorescent. And there's just examples of one type here, which has a fingerprint profile under EM. Each Batten disease is clearly inherited disease, clear monogenic inheritance, and all except one are autosomal recessive. And at the moment, these diseases are incurable. So, what I'm going to do now, so the most common type of Batten disease is one called CLN3 disease, juvenile CLN3 disease, which begins with quite a rapid visual loss around the ages of 6 to 10, and then sort of gradually disturbance in speech, onset of seizures, and then a progressive decline of cognitive and motor functions, and also accompanied by psychiatric signs, sleep disturbances, extrapyramidal signs, and eventually leading to death. And this could be in the late 20s. So here are 2 patients with juvenile form up here. So Laura, who's at the very early stage, and then this gentleman who is more advanced. One of the more rapid types is CLN1 disease. So here's a girl who is at 1 year, looks perfectly healthy, but 6 months later she can't even sit up without being helped, and a few years later is very severely affected. This is a group of families with their children, and most of these children will have CLN2 disease, which has an onset between 2 and 4 years. So over the years, and this was my background, we have identified quite a number of genes now that cause Batten disease. So in the beginning, of course, these were the most common types, and then more recently these have been genes identified in single families. And along the way, I set up a mutation database, which if any of you do come across Batten disease patients, you might find helpful. So this is hosted by UCL. It's part of a bigger resource website, but in terms of the databases, these are tables put together for each gene, summarising patients or the mutations separately, and they're trawled from the published literature or information supplied by diagnostic labs or even sometimes families themselves. And we try and present the data in a they're just Excel tables, so you can download them and manipulate them how you choose, and it can be helpful, for example, looking to see what mutations are particularly common in a country that you might have to consider in terms of diagnosis. However, what has been extremely useful from this is that when we look at all the mutations and the phenotype associated with those, that we see really wide disease phenotype variation, as of course is increasingly recognised with many diseases now. So just for example, looking at the CLN10 gene, which is the extremely early onset, very rare, so complete loss of function of this gene, which encodes cathepsin D enzyme, has a congenital onset, but there are also cases with mild mutations where onset may be delayed by a few years or even up into sort of late teenage, early adulthood. And that applies for most of these genes. We also have a few mutation-specific phenotypes, so particular mutations cause a disease that wasn't initially recognised as an NCL. So, for example, CLN8 gene, the first mutation, or rather the gene was first identified in a group of patients with a disease called progressive epilepsy with mental retardation, or northern epilepsy, which wasn't considered to be an NCL at that time. And then very recently, we're now seeing in quite a lot of the genes some very late onset disease. So CLN3, which I'll refer to later, we're now seeing adult patients who are only blind, and their mutation is found in CLN3. So this genotype phenotype is really broadening. Okay. Right. So, number of genes. Is there a common theme? So this was just a schematic to show you where the location of the proteins encoded by these genes are, and you can see that there's quite a few genes that encode enzymes within the lysosome, or even transmembrane membrane proteins of the lysosome. So that fits with the storage material that accumulates in the lysosome. And then, but we also have a few genes that are encoding ER membrane proteins. So somehow there's a connection in this disease between what's happening in the ER and the lysosome. Now, whether that's via a trafficking route or whether it's via direct organelle contact, we're not too sure yet. Okay. So back to BACCURE, and what I thought I'd do now is give you some examples of some of the work that we've produced through this project. So we had one work package that was providing new models and tools, and then a lot of interconnected work packages really focusing on, as I say, the biology and the basic background to the therapeutic development. So just to dip into some of these work packages. So work package 1, which was called New Models. There are quite a lot of model organisms available for Batten disease. Some of them are engineered, some of them are naturally occurring. So, for example, there are naturally occurring primates and sheep and dogs that have mutations in genes that cause Batten disease in humans. But there are also some lab models as well. And within BACCURE, we chose to focus on yeast and zebrafish and mice. So yeast as the simplest organism possible to study the genes that are conserved down to yeast. A few of them are for Batten disease. Zebrafish as a simple vertebrate that allows for some very fast screening or testing of drugs and also genetic manipulation. And then the mice for developing the gene therapy and testing the effects and also understanding more about the disease. In terms of developing new tools, LATAT here have been working on antibodies. It's been quite a challenge, but we have some. And we also have a lab that was developing iPS cells as well. So that sort of was the basis of the model work package. Now, the second work package, Pathway Lead, was an enormous work package coordinated by Juan, who's sitting right at the back there. So we always had to give a lot longer for any presentations within this work package. So I've tried to summarise it very briefly here. So what we were trying to do within this work package was understand more about the basic biology and gene function to provide in lots and lots of different ways. So genetics, cell biology, metabolic approaches, proteomic and bioinformatic. Then from that work to identify specific targets that we thought might be responsible for some of the defects that we were seeing within neurons in the disease, and then also to use that information to look at the effect of new compounds or test compounds that we might be studying during the process of the project. And obviously this work package was receiving information from some of the others, so of course from the new models developed, but also generating information that went out into other work packages. Excuse me. So just to highlight some of the work we've done using yeast as a model organism, because this is quite unusual if you're looking at a brain disease to think about using yeast for this. And what we did here was to try and do a very sort of take a systematic approach and look and identify genetic interactors with the conserved gene in yeast. So the CLN3 gene is conserved down to yeast, highly conserved, and we know that yeast lacking the equivalent gene, which is called BTM1, they have a plethora of phenotypes, and they can be rescued by expression of the human gene. So what we did here was do a synthetic genetic array approach where we deleted all the non-essential genes in yeast in the background of a yeast also lacking the equivalent CLN3 gene. And then there are a number of approaches we can take with the hits that we find. So we can look for differences between the different strains that we used. We also compared different disease-causing mutations as well. So we had quite a lot of information to deal with. We can look at things that overlap between them and significance, strongest interactors, and things that are common between them. So, of course, this is a lot of work and will continue beyond the end of the project. Now, in terms of work package 2, and trying to understand about the metabolic reprogramming that became apparent in the CLN7 form of Batten disease, where there was clearly some damaged mitochondria and some metabolic rewiring that was going on. So this is work that Juan is unpacking and is very interesting, and of course may help us in terms of understanding which drugs are going to help the CLN7 type of disease. Another lab from Germany led by Stefan Stork, we're looking at the disturbance of lipidomics and proteomics, and here just showing the results they've generated for the CLN7 type. So looking at sort of depletion of quite a number of the soluble proteins within the CLN7 lysosome. They've also found impaired mTOR reactivation in fibroblasts from the mouse model compared to the wild type, and impaired tubulation from the autolysosomes in the CLN7. The CLN7 gene, if you remember, is one of the lysosome membrane proteins. And when they look in the lysosome, either you're looking at the brain or, again, using the mouse fibroblasts, they can see an increase in certain lipids, and they can see, again, the decrease in the enzymes within the lysosome. So starting to understand changes, and some of these changes, of course, are going to provide useful biomarkers. When we have treatments, we'll be able to see whether the treatments restore the level back to what we expect for wild type, or whether actually it may indeed rescue, but it may produce a new normal. Okay, so moving on to sort of thinking about the drugs and development of drugs. We took two approaches for screening for compounds. One was screening, this is a lab in Italy, led by Diego Medina, used human cells and did high throughput screening on human cells. And, we also, again, made use of the yeast and did high throughput screening to identify compounds that rescued yeast. And again, we can, of course, test yeast that model different mutations in the disease gene, and then also test the compounds on yeast that model other diseases, other lysosomal diseases beyond Batten disease. And then, of course, take those compounds into patient fibroblasts and eventually into testing in vertebrate animals. So in terms of therapy, as I say, we decided to focus on gene therapy and drug approaches, and we're aware that we have to target the brain, therapy to the brain, to the eyes, and then we also have to think about the rest of the body, because the storage material occurs throughout the body, although the disease that we recognise is really a brain disease. But of course, if we're able to improve what's happening in the brain, it may be that other problems arise during the rest of the body, in the rest of the body. So to show you some of the gene therapy work, we've first of all been developing eye gene therapy, and we began with CLN6, and just very briefly, in a mouse model that's untreated, at 9 months, the photoreceptor layer, which is here, is severely reduced. So from there, the number of photoreceptor rows is very severely reduced, but with a gene therapy, we can restore the rows of photoreceptors here, which you can see visually here. But what was really interesting about this was in all other eye gene diseases that have been targeted by gene therapy, if you put the gene into photoreceptor cells, that's what worked. You stop the photoreceptor cells being lost. But for this one, if we did that, the photoreceptor cells still died. We actually had to target the gene therapy into the middle layer of the eye in order to prevent the loss of the photoreceptor. So this was a novel finding completely, and obviously we've now been aware of that as we've gone forward with some of the other gene therapies for the eye for the NCLs. And then here, Ahad Rahim has been leading on gene therapy for the brain, and again, just to show you one result of the CLN6 mice that would normally die by a year, but with the gene therapy into the brain, we're extending the treatment to 2 years, which is about the length of the same as wild type mice. So this is working very well as well. Okay. Now, if we're going to move forward with therapy, you do and clinical trials on rare diseases. The approach that's been taken is to compare the effect of any therapy against the known natural history of the disease, rather than having a control group that isn't treated at all. And the reason we can do this for Batten disease is because of the Child Disease Registry, which I mentioned earlier, which has now become an international disease registry where a lot of information is now collected for all the different types of Batten disease. There have been a number of clinical trials over the years for the NCLs, most of which have had little benefit at all. There's just one that's been successful, and in terms of the 3 that we're working on, the 3 CLN3, 6 and 7, there have been some very recent trials started in the USA with gene therapy where they're really just trying it out. So it will be years before we know the result of this, but what we feel we're doing within our mouse gene therapy work is really providing some of the science behind gene therapy to really understand what's going on and what the key things are that we need to be aware of. Okay, just one of the highlights from the consortium was we ran an international meeting last year in London, and so there all the people that sort of gathered from around the world working on Batten disease, and yeah, that was a good outcome for us. I've already mentioned the fact that we want to produce the 3rd edition of the textbook. And I'll close by just showing you all the different logos of the labs that have been involved in this disease, in this project. And I'd be very happy to take some questions. Okay.
No, yo soy Gustavo, soy el papá de Marta y María, que tienen una enfermedad, bueno, tienen Batten CLN6. Sí, tienen Batten CLN6 y están participando en el ensayo clínico que hay en Estados Unidos actualmente de terapia génica. Con la doctora Emily de los Reyes muy bien. Entonces, pues bueno, llevan la mayor lleva 1 año en el ensayo clínico y la pequeña 6 meses. Quería preguntar pues, 2 cosas. La primera es si todas las vías de investigación que se están llevando aquí a nivel europeo pueden ser complementarias con esa terapia génica que están, pues eso, tratando a mis hijas ahí en Estados Unidos. Y la segunda pregunta es, no sé, es muy complicado encontrar a familias que están en nuestra misma situación, sobre todo pues con la enfermedad del tipo 6 que tienen nuestras hijas. En España no hemos encontrado, hay unos gemelos que la tienen, pero no hemos podido contactar con ellos y no conocemos de nadie más. Thank you so much.
Yes. I can help you with the second question about the vision. Okay, so first of all, to answer your question about the therapy, so the principle of the gene therapy in the States and what we've been working on is very similar. And we've been trying to we're trying to think about how early you need to start this therapy, thinking about dose, thinking about sort of the details of it. So going beyond what was known before the trial started. And so I guess all we can say is certainly for in mice, which he will know about and which was done in the States too, that in mice the gene therapy seems to work well. But of course we don't know what it's like in a child, a larger animal, you know, or a child. Okay. Okay. So, yeah. So in terms of getting in touch with families with the same mutation, a lot of that seems to be done through social media now. But of course, we will know from the mutation. We can look in the database to see approximately how many families we're aware of and where they're from. So that could help target families, perhaps. I.e., is it a Spanish mutation or is it throughout the world?
Solo decirte que en España tenemos dos grupos de neuropediatras que estamos trabajando con enfermedades lisosomales y estamos haciendo una base de datos y estamos recogiendo todos los pacientes con los diferentes grupos, entre ellos las lipofuscinosis, pero que no siempre todas las familias quieren contactar con otras familias y eso es algo que nosotros también tenemos que respetar. Nosotros siempre les decimos que hay posibilidad de contactar con otras familias, la mayor parte de ellas están de acuerdo, bueno, algunos no lo están tanto, ¿no? Y sobre todo los que viven en sitios más periféricos o que no van a centros de referencia todavía, eso son más difícil de contactar con ellos. Por eso estamos haciendo un trabajo de búsqueda con todos los médicos neuropediatras españoles. Sí, luego.
Nosotros estamos haciendo distintos eventos para recaudar dinero, pues para ayudar en la investigación. No sé si sería posible el poder también pues contactar con ellos para pues bueno, si lo necesitan, si es viable. Ajá.
Sí, podemos, en este caso, en la pausa. 4 menos cuarto, podemos hablar y coordinarlo todo, porque de hecho el foro es el adecuado, o sea, aquí hay varios investigadores de varios centros, de varios hospitales, creo que es el foro adecuado y conseguiremos una forma a ti. Okay, any more questions? I think we have some for 1 or 2 more. If not, I think we are in excellent time, so we can move on. So I'm so happy to introduce Dr. Martinet from Minorix Therapeutics.